Hi, I'm Max Avenido,
I'm Tsoler Demerdjian.
and we're in the Martin Lab, and we're presenting on in vitro expression and purification of human gamma S crystalline proteins, which are in our islands used for structure, but when exposed to UV, they can aggregate and cause the lens to be cloudy, which causes cataract. And what we did was we expressed and purified the wild-type version of these crystalline proteins.
So we first started with transformation, which is where we introduced the bacterial DNA into a competent cell. And then we went into expression, which essentially produces a large volume of that bacterial cell, including the gene of interest that codes for our protein, the human gamma S. And then we went and purified it, which essentially breaks down that bacterial DNA until all we're left with is just our purified protein, which can be seen in our results. From expression all through the different steps of purification, it gets more purified until there's just one distinct band, which is where our protein is.
And then we also did a mass spec to be more sure that we actually have this purified protein. And we were just looking at the main peak of our experimental to the expected peak, and they do line up. Although these peaks are kind of also more obvious in our experimental, we're thinking that it might be due to low yield of the protein. And then for the future, hopefully that these wild-type proteins can be compared to other proteins that are being expressed by other projects, and then therefore we can also create a more accessible treatment for cataract for lower socioeconomic, for like historically marginalized communities.